If we brought 600 trillion bacteria with us, how do we sort out the good ones from the bad ones? And if some of them are producing toxins, that's not such a good thing for keeping us healthy as an organism. So how we think about all the bacteria that live with us is a very complex question. And our next investigator, João Javier, is going to come and tell us a little bit about how he uses computational tools along with wet lab biological experiments to try and resolve some of these issues. João, it's yours.
All right. Thank you very much, Greg. Okay, so I'm João Xavier, and I'm in the program for computational biology. And we also have wet labs, so we do both computational and wet lab experiments. And I'm going to talk about a few topics here that I hope will unify some of the topics brought up by the previous colleagues here, which is that when we look at that diversity of organisms that we presently have on Earth, one thing that really pops to my eye is that there are different levels of biological organization. So there are prokaryotic organisms like the bacteria. Then we have eukaryotic single-celled organisms, single-celled microbes that are eukaryotic. Then there's multicellular organisms such as ourselves, which are made of many, many different cell types. And then we even have societies of organisms, like the colonies of ants and bees. And what really characterizes these different levels of biological organization is that in order to go from one level to the next one, you have to have evolution. Of course, that's really the driving process of all diversity that we have on Earth. But also, you have to have evolution of cooperation. So some individuals at the lower level have to come together to form a higher level of selection. And of course, even the first prokaryotic cells had to come from somewhere. We don't know exactly what that was, but some kind of self-replicating molecules had to come together and form the first prokaryotic cells. Now, when you go from bacteria, so from prokaryotic organisms to eukaryotic organisms, you have to have assemblies of prokaryotic organisms that form a larger cell, like the mitochondria that Cole talked about. So when mitochondria get associated with other cells, they form large eukaryotic cells. Then to go from eukaryotic cells to multicellular organisms, you have to have many cells come together and form an organism. And then to form societies like colonies of ants and bees, you have to have many animals come together and form a larger society. So you see the same pattern of elements coming together and forming a larger community over and over throughout this large biodiversity that exists presently on Earth. Now, the same way that you have cooperation evolving, you also have competition among the units that form a community. And cancer is one example of this type of competition. So cancer is a disease that happens inside the body where cells start competing against each other rather than cooperating. And we want to understand these type of processes. When do competition and cooperation come together to cause problems? And the same way that we can use simplified models to understand development, like Kat mentioned, she can study embryonic development using mice. And Cole mentioned how we can use worms to study a function of mitochondria. We can also use colonies of bacteria to study how cells come together and form larger assemblies. And we can study a process very similar to cancer that occurs in these colonies of bacteria that we call evolutionary cheating. This means when bacteria come together to form communities like biofilms, and I'll talk a bit about that, there's always the propensity of certain cells to misbehave inside that community and just exploit the resources that other cells are producing. And these we call cheaters. We're also very interested in associations between different levels of biological organization, like the microbiomes. I'll talk a bit about that at the end of my talk, which is basically associations between bacteria and the human body. And in many instances, these bacteria that live in and on our bodies that really outnumber our own cells, the own cells that we carry in our body at a ratio of 10 to 1. We have 10 times more bacterial cells than we have cells of the human body. They're really important for our health. And we want to understand how we live with them in peace, but sometimes things can go wrong. Just like they happen in cancer, things can go wrong with our microbiomes, and they can end up causing disease. Okay, so I'm showing here a Petri dish, which is 9 centimeters wide, so it's about this size. And on that Petri dish, we have a colony of the bacterium Pseudomonas aeruginosa. So the same bacterium that Cole talked about that he uses as a model pathogen in his experiments. So here, we're using Pseudomonas aeruginosa as a model for collective behavior in bacterial cells. So I also produced here a time lapse. So we're going to see how this bacterial colony developed over time. We sped up this so that 24 hours in real time actually passed by in 30 seconds. Okay, so we'll see at the beginning, we put bacteria at the center. They're growing and dividing. They form a colony. And then you see these tendrils shoot out of the main colony, and they migrate towards the edge of the plate. Now, each of these tendrils here is made of billions of bacterial cells. Each bacterium cell is about 1 micrometer long, so we can't see it by eye. But together, they can achieve really strength in numbers and migrate towards the edge of the plate. Okay, and they can do so in the time scale of a few hours, which a single bacterium cell could not. So by coming together, they make these huge communities, and they can really propel themselves across the plate and migrate distances that are impossible for a single bacterium cell. So this is real cooperation among bacteria. Okay? So to be able to migrate on top of the Petri dish, the bacteria, they need to produce, they need to synthesize. Each bacterium needs to synthesize large amounts of biosurfactants. So these biosurfactants are detergent-like substances that the bacteria produce. And once they are secreted out of the cell, they coat the surface of the Petri dish. So they literally lubricate the surface of the Petri dish, and that allows the colony to slide on top. This is, again, a cooperative effort, because if you have just a single bacterium producing these biosurfactants, they won't make much of a dent, right? So it doesn't accumulate that much, and it's not enough for the bacterial colony to slide on top. But if there are billions of bacteria producing these surfactants, then they can start coating the surface, and now they can migrate. And we can see here, this is a really zoomed up version of the bacterial colony. Now, this bar here represents 200 micrometers, and this was taken using a microscope. And here's the edge of the bacterial colony. We still cannot see individual bacterial cells because each one of them is just 1 micrometer long, so it's 1 to 200th of this bar here. But we can see the layer of the biosurfactants that the bacteria are producing. And when I start this video here, you see the layer of the biosurfactants advancing, and the bacterial colony is migrating behind it. So first, they pump out these massive amounts of biosurfactants. Each bacterium cell is contributing with its own small fraction of this, and then collectively, they can really coat the surface and allows the colony to slide on top. Okay. Now, these bacteria are prone to cheating. So here I have in the same Petri dish bacteria that can swarm, so they produce biosurfactants. And here are cheater bacteria, which are mutants that we produce in the lab, and they can't produce their own biosurfactants. But if they get mixed with the wild type, they can use the surfactants of the wild type to migrate. So here in this experiment, we had labeled the bacteria with fluorescent markers. So the mutant was labeled in green, and we could really confirm that they're migrating by using the surfactants produced by the wild type bacteria. So this is a form of cheating, of social cheating, of free riding, where basically these mutants are just waiting for the wild type to make all this effort of producing biosurfactants. They just sit around doing nothing, and when the biosurfactants arise, they can free ride on the biosurfactants produced by the wild type and still propagate themselves across the plate. Okay, so now what we found over the course of a few years is that the bacteria, Pseudomonas aeruginosa, they actually make a very well-informed decision on whether it's time to produce surfactants. And they do this very well-informed decision because they want to avoid being cheated on by mutants that don't produce the surfactants. So here in this plot here, I'm summarizing the results of several years of our research, where we really figured out that what these bacteria do when they start expressing genes for biosurfactant production is that they really measure how many bacteria are already in the colony. So they're basically asking how many of us are in the colony, and does it make sense to start producing surfactants? Because if I'm alone, it doesn't make sense to produce the surfactants. I won't go very far. But they also use in their decision a measurement of whether they can afford, as individuals, whether they can afford to produce the surfactants. Basically, do they have enough carbon and energy source that they can afford to produce the surfactants without that slowing down their growth. Because if they produce it too early, and that's going to slow down their growth, they might be outcompeted by cheater mutants that don't do this decision well. Okay, so this is another way to summarize it. The bacteria, at the beginning of the swarming assay that I showed you in the previous slide, they have a lot of nutrients, both carbon and nitrogen sources. And at this time, they don't produce surfactants. They just grow. After a while, they run out of nitrogen source, but they have excess carbon. At this point, they can really afford to start producing surfactants because there's way too much carbon, way more than they can use for growth. So they can afford to produce surfactants, and that allows them now to slide and go and hunt for more nutrients elsewhere. Okay? So we call this feature of bacteria, we call it metabolic prudence, because we believe the bacteria are doing a very prudent decision on whether they can cooperate by producing surfactants. And this way, they avoid being cheated by free rider mutants. Now, in order to dissect this type of cellular decision-making, we use a novel type of thinking in biology that is called systems biology. So what is systems biology about? Systems biology is about understanding how molecular circuits inside cells, so molecular processes like gene regulation, work together to enable cells to make cellular decisions. One example of this is the ability of cells to understand whether they're in a population. And we call this process quorum sensing. The way quorum sensing works is that each bacterium can produce a certain amount of an extracellular molecule. So they start pumping out small signaling molecules that accumulate in the extracellular space. And that allows the bacteria to sense, by measuring the extracellular concentration of this molecule, whether they're in a low-density population, like this one here, or in a high-density population, where it's very crowded and this molecule really accumulates in the extracellular space. And just to illustrate how this works, think of this situation where you're locked in a dark room with a bunch of people, but you don't know how many people there are in the same room as you because it's dark. But if everyone starts shouting, by listening to the volume of how loud it is, you can more or less evaluate if you're alone in the room or if there are many people together with you. And so this is what bacteria do in quorum sensing, is they produce these signaling molecules, and then they listen, and listen here is between quotes. They're not actually listening for sound. They're measuring the extracellular concentration of this molecule, and if the extracellular concentration is very high, that means there's a lot of bacteria present. If the extracellular concentration is low, that means that bacterium is probably alone and doesn't make sense to try to swarm if there are not enough cells. Now, in systems biology, what we do is we recognize systems such as quorum sensing, and we try to identify those type of systems in different biological systems. So, for example, quorum sensing, we can find different types of species of bacteria that have quorum sensing systems, even using completely different molecules. We find similar systems in tissue morphogenesis in order to form certain types of tissue patterns. Cells produce signaling molecules. And also in the immune system, and we also see examples in metastasis. So as systems biologists, we're trying to understand patterns of how cells regulate different types of behavior, and we want to find these across different type of multicellular systems. So in order to understand that, we really need to study across different types of models. So I mentioned that we work with bacteria. We also work with cancer biology, and then the human microbiome. And so now I'm going to describe a bit our work in the human microbiome, where we look at many different species of bacteria that live in and on our bodies and that are important for our health. So this diagram here intends to represent the different type of communities that together make up the human microbiome. So we have an oral microbiome, we have a skin microbiome, a lung microbiome. The most important in terms of numbers, actual numbers of bacteria, is the gut microbiome. So these are bacteria that live in our gut, and they help us digest our food. Okay, so in addition to helping us digest our foods, the bacteria in our guts also have a very important secondary role, which is they keep pathogens out. Because if these bacteria weren't there, then the way would be just open for other harmful bacteria to come in and colonize. And so these bacteria that naturally colonize our guts and help us, they also ecologically keep other pathogens out. So we want to understand how that actually works, how the gut microbiota confers resistance against invasion by pathogens. And then we want to understand the harmful effect that some antibiotics have in the gut microbiome, where they end up harming the good bacteria, and then they open the way to invasion by pathogens. And then also, at the bioinformatic level, we want to develop mathematical models that will help us predict how the gut microbiome is going to change over time, pretty much like we use mathematical models to predict how the weather is going to change over time. So the gut microbiota is a very complex system, just like the weather is. And so by developing mathematical models, we hope to be able to predict a bit in the future how the microbiota is going to change, for example, in the context of a patient that's being treated with antibiotics. So here's a summary of why bacteria resist colonization by pathogens. So we have here a representation of the gut microbiota. So it's a very diverse system. It's like a zoo. You have all these bacteria cooperating with each other, producing metabolites, and they interact with the immune system. And this ecosystem resists colonization by pathogens. For example, Clostridium difficile or vancomycin-resistant Enterococcus. There are two types of pathogens that we're very concerned about in fragilized patient populations, like bone marrow transplant patients here at Memorial Sloan Kettering. Now, when patients take large doses of antibiotic, that kills a lot of the bacteria, the good bacteria that we have in the gut, and opens the way to colonization by pathogens. So these pathogens have their way free to come in and cause harm. Here I'm showing you data from an actual patient here from Sloan Kettering. This is data from a bone marrow transplantation patient, and we looked at the microbes that they have in their gut by analyzing their stool, so poo samples. And we have here, poo sample 9 days before the transplant, and these different bars here, these different colors, they represent different bacteria that were present in the gut of this patient. So there's many different colors. That means the gut microbiota was healthy and very biodiverse. And then here, we have the day of the transplant, and up here, we see these bars representing antibiotics that were given to the patient over time, okay? So as the patient approached the bone marrow transplant day, the doctors start giving the patient antibiotics, which are really necessary to prevent diseases. But then we see that one single bacterium here starts dominating the gut of this patient 6 days after the transplant. And this bacteria caused real harm. So it caused a bloodstream infection, a bacteremia. It was actually vancomycin-resistant Enterococcus. And then later on, the doctor stopped vancomycin, and this disease went back down. But then later on, the patient became more fragilized, and vancomycin-resistant Enterococcus was starting to increase again in abundance in the gut of this patient. So we want to understand how a biodiverse microbiota, like this one here, prevents expansion by a harmful bacterium like the vancomycin-resistant Enterococcus, and how do antibiotics cause this problem, and how we can better understand this process so that we can prevent it from happening within patients. Another way of thinking is think about the microbiota composition as being here, this ball, and it's sitting on a valley, here associated with a healthy microbiota. And if we perturb that system by, for example, changing our diet or taking a mild dose of an antibiotic, it still stays in a healthy state. But if we give very, very strong doses of antibiotic to a patient, that microbiota composition may end up in a disease state that is itself very stable and very hard to get out of. So by developing mathematical models, we want to understand how many different states there are, how do you go from healthy to disease and back, so that we can design novel therapies that keep pathogens in this healthy state. Okay? So here's an experiment that a graduate student, Charlie Buffy, carried out to evaluate this with mouse models. So he took a mouse, and he gave this mouse a single dose of an antibiotic called Clindamycin, and then he evaluated the effect that that antibiotic had on the gut microbiota of this mouse. Then he did another experiment where he gave the same antibiotic to another mouse, and then he inoculated this mouse with a pathogen called Clostridium difficile. So here he's trying to evaluate if you perturb the microbiota with an antibiotic, does that make it possible for the pathogen to colonize the mouse and cause harm? And here he had what we call a control experiment, which is a mouse that did not receive the antibiotic, but received the pathogen. And so we want to compare these two. Does the pathogen succeed better after the mouse received the antibiotic? Okay? So here we have what we call a survival curve. Here the x-axis represents days after infection by Clostridium difficile. And here on the y-axis, we're showing the number of mice that survived. And what we see here in this black dashed line is the number of mice that really survive because they had intact microbiota. So these are the mice that did not receive antibiotic, and 100% of them survived even though they received the pathogen. Whereas the mice that receive antibiotic and then the pathogen, so this red bar here, 40% of them died within the first 5 days. So this is a really good model to understand that by giving antibiotics to a mouse and then giving them the pathogen, we really increase the mortality of this mouse because of this infection. Now, what we do also in the lab is that we analyze the composition of the gut microbiome using novel advanced techniques of DNA sequencing. So these are very, very advanced techniques that allows us to really understand which microbes are there and how abundant they are. So this is a very busy slide, but it's a slide that summarizes the experiments that I showed you before. So here, we're representing data that comes from the mouse, the control mouse, that didn't receive the antibiotic, just received the pathogen. And these different colors here represent abundances of the different bacteria. So the different bacteria are these rows here. And the fact that we see a lot of colors means that the microbiome is very, very diverse. And it stays very diverse. And the gray here represents absent bacteria. And here, the pathogen, we never see it colonizing the mouse because there's all these other bacteria that are keeping the pathogen away. Now here, we have the experiment where the mice received the antibiotic. So this is before the antibiotic. The microbiota is very diverse. Then after the antibiotic, most of the bacteria are gone. So the biodiversity of the microbiota drops. It's like it's opening the way to invasion by a pathogen. Now, we didn't give pathogen to this mouse, but we gave it to this mouse here. And so here, it's before the antibiotic. It's very biodiverse. After the antibiotic, biodiversity goes down. And then we colonize the mouse with the pathogen, and we see the pathogen creeping up and colonizing the gut of this mouse. So it's really showing that a single dose of clindamycin, the antibiotic clindamycin, can clear a lot of the biodiversity away from the mammalian gut and open the way to colonization by pathogens. Now, as we discover that the microbiome is really essential for human health, for example, to protect against pathogens, we're also discovering new features of the microbiome. And one of them is that it really is important to control behavior. Okay, so this is a new topic of research in the field of animal behavior, but the evidence is mounting that the microbes that colonize animals really influence behaviors, different types of behaviors of different animals, and the behaviors that animals have also influence which microbes get to colonize. So this is a very interesting topic of research that's really being propelled by novel technologies like high-throughput sequencing. Okay? So in this slide here, I'm showing here in the x-axis years, right? So this is time. And here on the y-axis is the price that sequencing costs. And the green line here, we're showing that the price of sequencing, high-throughput sequencing that comes from novel DNA sequencing technologies, is dropping very, very rapidly. It's actually dropping faster than Moore's law. And Moore's law explains how cheap computers get over time, right? So we know that if you buy an iPhone today, it costs much less than the same iPhone 5 years ago. And the reason for that is Moore's law. So if you look at, you apply this same sort of thinking to DNA sequencing technology, you see that DNA sequencing is getting cheaper and cheaper, which enables scientists to do research much, much faster and in casting much wider nets to explore different biological problems. Just to put this back into perspective, let's think about how computational power advanced so rapidly. And here we have a calculation rule, which I don't even know how to use, but it's the main tool that my father used when he was in engineering school in the 60s. So this is how they made calculations. They didn't have computers at the time. Now, when I studied engineering in the 90s, I had this calculator here, which was pretty good at the time. And then now, when my student, Vani, is teaching bioinformatics students, these days, most of the students will have something like this, as I imagine most of you will. And some of us will also have something like this in our pockets. So you can see over the years, machines get much, much faster and much, much cheaper. And something like this that was inconceivable 30 years ago is now a reality, and its price drops very, very rapidly. So now what happens when you try to apply the same type of thinking, and you think that DNA sequencers right now, which are only a reality in research institutes like Memorial Sloan Kettering, if its price drops very, very rapidly, it can really advance and make DNA sequencing more affordable, and eventually you can have some sort of personalized DNA sequencing that we can all use, for example, to analyze our gut microbiome in our own homes. Okay, so maybe this is still science fiction, but we hope that this type of DNA sequencing technology can become a reality within 5 to 10 years, so that it can be used in clinical settings to analyze patient data such as these. Okay, so thank you very much for your attention, and I'm happy to answer questions.
Okay, go ahead. Let's keep it quiet for the questioners.
Hi. You said that the old DNA sequencing technology, it changed sort of, and that created a price drop. What exactly characterized that change in DNA sequencing?
So what makes DNA sequencing much faster these days?
Yeah.
There's part of it is technological advances. Part of it is commercial interest, that companies suddenly became very, very interested in this. And the fact that there's competition among companies to make the latest technology is really driving the field forward very, very fast. So it's a combination of factors. But the reality is that its price is becoming much, much cheaper, and that we have to take advantage of it.
Over there.
During quorum sensing, when the cells and the bacteria are trying to get a sense of how many there are in the colony, can they sense which one are the mutated bacteria so they can prevent the cheating? Yes.
So that's actually a topic that we're very, very interested in finding out. We don't know the answer to that entirely, right? So she asked when we have a cheater mutant within a colony, can the other bacteria recognize that there's a cheater mutant by, for example, monitoring the types of quorum sensing signals that they're producing? The answer is we don't know. We predict that this would be a good way for bacteria to prevent cheating. If you could recognize that another bacteria suddenly became a cheater, that would be really, really important. And for sure, that does happen in the human body. When there's cancer cells, other cells can recognize that there's a cancer cell and try to eat it up, right? So we think there might be something similar happening also in bacterial colonies, yes.
Great. Over here.
So when the antibiotics are taken and the sort of good bacteria dies off, how does the body regulate how much of the good bacteria is regenerated, or at what rate is this done?
Yes, so again, an excellent question. This is also an active topic of research. It's still, we don't know the right answer. What we do believe happens is that the human body can, in some way, sense how we're colonized. If we're colonized by a lot of bacteria, what is the sort of, in broad strokes, what is the composition? Not exactly which bacteria there are, but more or less if they're good bacteria or bad bacteria, and can produce substances to try to control the composition. What I mentioned also about the behavior, which is also very interesting, is that we believe that the gut microbiota influences behavior of humans and also other animals, for example, by generating cravings that give you certain types of appetites that might be connected with trying to control the gut microbiota composition.
Over here.
So you mentioned that the problem with current antibiotics is that they are killing all of the bacteria, good and bad, inside the gut.
Mm-hmm.
So would it be possible sometime in the future to create antibiotics that target specific bad bacteria?
Yeah.
in order to just clear them out? And is it something your lab is interested in working on?
Yeah, I mean, that's an excellent question again. Part of the problem indeed is that we developed, our first line of defense against bacteria are antibiotics, and they have different types of spectrum, right? So some are broad spectrum, which kill indiscriminately a lot of bacteria. Some are narrow spectrum that target some of the bacteria, but still all the effort in developing antibiotics has been towards thinking about let's kill bad bacteria and not thinking about their effect on good bacteria. And of course, the next line of thinking will be to target specifically bad bacteria. Now, a sad reality is that the commercial incentive for developing new antibiotics has decreased. So pharmaceutical companies are putting less and less effort to developing new antibiotics. This is a sad reality. It's something that we need to change. And possibly in academic centers like here, we can hope to develop new cutting-edge drugs that will target specific bacterial species, harmful bacterial species.
Thank
Right
you.
here.
Hi, yeah, I had like a very similar question. Do you think you could create some sort of antibiotic that could almost work with the bacteria like in the gut, so it could sort of protect it instead of killing off other bacteria?
Some kind of beneficial antibiotic that would not harm the bacteria in the gut, but would harm pathogens.
Yeah.
So, I mean, the answer is similar. Of course, we hope we can do that. Unfortunately, pharmaceutical companies are not so interested in that right now because it doesn't, according to them, doesn't seem to be a good commercial path. But I think academic centers like Sloan Kettering, which are beyond commercial pressures, can invest in this type of strategies for sure.
Over here.
Yes. When they're sometimes, for instance, like dogs or humans, they're infected with those types of, like, for instance, ringworms in the stomach and the intestine. And you said when you use the medicine to cure the pathogens, I mean, medicinally, it's very expensive to purchase those the medicine which will can cure these worms. But how do those worms specifically infect the intestine, like similar to those pathogens, and can they be cured with the same type of medicine in the stomach?
Okay, so I mean, antibiotics, they target only bacteria and not those worms, but of course there will be specific drugs that will kill other types of parasites. They're potentially the drugs that veterinarians would use to kill parasites like those will not affect the microbiome of the animal so much. So at least that part of the problem shouldn't be as important in a situation with parasites that are not bacterial. And so I hope that problem won't be so important there. I hope this answers your question. Maybe we can discuss a bit more, because I don't think I understood all the points of your question. Yeah. We can discuss
Just a couple more questions, and then all the speakers will be down front for specific questions after. So we'll take one more on each side.
Hello. I'm curious as to, like, do bacteria from cooperative environments such as the gut microbiome, do they behave differently when they're taken out of this cooperative environment and put into a petri dish?
Yes. That's an excellent question. Did you have a follow-up? No. Okay, I'll answer that question. That is an excellent question, because when we take bacteria out of the gut, most of the time they won't survive. And that has been a strong impediment to developing the field of the gut microbiome, because scientists don't know how to culture the bacteria that we have in our gut. Once we bring them out, they die, right? So it's only now with high-throughput sequencing that we can analyze the bacteria directly without having to culture them. So we analyze their DNA and we know which bacteria are there, that we can finally study them. And this is a development that only occurred, for the past 6 years or so.
Last question.
In the mice that were exposed to pathogens after, there was a pretty high mortality rate in the mice that were exposed to pathogens after being treated with antibiotics. And obviously they were intentionally exposed to pathogens. But I was just wondering, is there any sort of significant mortality rate associated with people who were treated with antibiotics?
We do observe very increased risk of infection by pathogens in patients after taking certain types of antibiotics. So we observe very high incidence of bacterial infection. Most of the times, of course, patients don't die, and that's, of course, great. But they do get the infections, and they're life-threatening.
Well, I want to thank all three of the speakers, so we give them a round of applause. And I thank all of you for participating, both here in the room and the outside rooms and are on the internet. We really hope you got stimulated by some of the science and the possibilities that you've seen here today. And please come back next year.